Assalamualaikum and hello everyone. In this demonstration video, we will be presenting size exclusion chromatography SEC. And this is our group members. And this is the content that we will be present in this video. What is SEC? SEC is size exclusion chromatography, that known as gel filtration chromatography, where it is a powerful analytical technique widely used in the separation and analysis of molecules based on their size. The stationary phase in SEC consists of porous space that typically made from cross-linked polymers such as tetran, agarose, and polyacrylamide. These beads have pores of different sizes that allowing smaller molecules to enter the pores and take longer to elute from the column. In contrast, larger molecules cannot enter the pores and elute more quickly, resulting in a size-based separation.
For sample preparation, we will discuss about goal of sample preparation, materials and procedure of sample preparation, and what the important considerations should be included. Proper sample preparation is super important to protect the column, get reliable results, and make sure your separation goes smoothly. Let's start with the goal of the sample preparation. There are 5 key goals preparation. First, clarity. We want to remove all the particles to avoid clogging the column. Second, solubility. The sample must be fully dissolved in the buffer. Third, compatibility. The buffer must match the column and mobile phase to avoid protein denature or unwanted interaction. Fourth, contraction. It should be optimized to give a strong signal without overloading the column. And lastly, degassing. To remove gases, prevent bubbles that can interfere with the detector of flow. These are the materials used during sample preparation SEC. Among them are your sample, such as protein extract, SEC compatible buffer, microcentrifuge tube, syringe filter, microcentrifuge, vortex mixer, or gentle stirring devices. Before loading the sample into the column, several preparatory steps are crucial. First, buffer selection and preparation. Choose a stable buffer such as phosphate buffer. Add 25 to 150 millimolar natrium chloride to prevent non-specific interaction and ensure the buffer is filtered and degassed. Second, sample dissolution. Dissolve polyphylized or concentrated samples in buffer, mix gently, and allow large molecule to hydrate for 12 to 44 hours. Third, adjust sample concentration. Aim for 1 to 5 mg per milliliter protein concentration or 0.5 to 4% of column volume. Then, clarification. Remove particulates using centrifugation followed by filtration to avoid clogging. Lastly, degassing. Use a sonification bath or vacuum degassing to eliminate a bubble that can interfere with flow or detection. Before running size exclusion chromatography, there are a few important things to keep in mind to make sure everything goes smoothly. First, sample stability. Make sure your sample is stable in the buffer you're using and that it can stay stable at the temperature of your chromatography run. Second, avoid too much dilution. We do need to clarify the sample, but try not to dilute it too much unless it's necessary, like if the sample is too concentrated. Third, the column care. Always check that your SEC column is properly stored and fully equilibrated with the same running buffer, and avoid sudden temperature changes because that can cause the air bubble inside the column. Last, watch out for contaminants. If your sample still has things like lipids, detergent or salt from earlier purification step, they can interfere with the separation or even damage the column. In that case, you might need to do extra cleanup like dialysis or precipitation before SEC.
Technical description. How SEC machine run. Firstly, size exclusion chromatography system have several main components. First, the pump which push the mobile phase, usually a buffer through the system at a controlled flow rate. Second, the injection loop where the sample is loaded using a syringe. It holds a precise volume ready for injection into the column. Third, the column which is packed with the porous space that separate molecules based on their size. 4. The detector. Usually, a UV detector records the molecule as they elute from the column. 5. Finally, the fraction collector collects the separated sample into label tube for further analytics. Next, here is how the SEC machine runs step by step. First, the column is equilibrated with the buffer to prepare it for sample separation and to stabilize the baseline. Second, the sample is loaded into the injection loop using a syringe. Third, the sample is then injected into the column by switching the valve to direct the flow through the loop. 4. Inside the column, separation occurs based on the size exclusion. Larger molecules are excluded from entering the pores and elute first, while smaller molecules enter the pores and elute later. 5. As the molecules elute, they pass through the UV detector, which record their absorbance and produce peaks on the chromatograph. 6. Finally, the fraction collector automatically collects the separated components into tube for further analysis. To gain a deeper understanding, let's watch this video clip.
Pumps to direct proteins containing solutions and buffers, so pH stabilized salt waters, through these different tubings, through different types of columns, And so with the size exclusion chromatography, we actually do it by, so there are different ways you can get your protein onto the column. So there's like sample pumps, which will suck up your sample and then pump it on. But with size exclusion chromatography, you want to have your sample in the smallest volume possible, because unlike affinity chromatography, your protein isn't actually going to get stuck to the column, and so that your protein is going to flow at a different rate depending on its size. This means that your protein is not going to get concentrated as it goes through the column. It's just going to get diluted. So you want to have it get diluted as little as possible, which means you want to start with the smallest volume as possible. So you start by concentrating your protein. We do this with these spin concentrators that we use the centrifuge, and so these tubes have a membrane, and you choose a size exclusion limit to make sure that the membrane's holes aren't big enough for your protein to flow through, because you want your protein to stay in the little membrane cup part. And then you spin it and spin it and spin it, and your protein gets concentrated, and then you can inject it onto this column. And so that little green tubing, those are sample loops, so you can choose a sample loop based on the size that you inject. And so you inject your sample, make it stored in the sample loop until you tell it to go onto the column. But the idea here is that you want as little volume as possible, so that your protein doesn't get diluted as it goes through the column. So what happens then is that your protein solution travels through the column, and then it passes through a UV detector. So proteins absorb UV light strongly at 280 nanomolar, thanks to the aromatic rings of like tryptophan, tyrosine, phenylalanine. And so basically this allows you to monitor when your protein is coming off, or what we call eluting, from the column. And the UV detector is in between the column and the sample collector. So as it passes through the UV detector and into the sample collector, you can then, it'll give you this graph called a chromatograph. And that's going to show you the UV and the elution time. And then you can cross-reference that to the different wells of the block, so you know which wells have the protein, and you can then collect it. So when you look at the chromatograph, the height of the peak is going to tell you about how much of the protein is there. And the elution volume, or elution time, so you can do it either, you can graph it either based on time or based on the volume. So obviously the faster you flow the column, that'll affect the time, but the volume will still be the same. And typically the slower you do slower time will give you a better resolution, but it can also take longer, obviously.
In summary, the SAC machine uses the pump system to deliver buffer. The sample is injected into the column where the separation occurs based on the molecule size. The detector records the pace, and the fraction collected collects the separated molecules.
SEC is an HPLC-based technique which contains porous particles in column to differentiate molecule in terms of size or molecular weight. The basic principle which applies to SEC is the movement of the solute and the solvent molecule across the pores in the stationary phase and their separation through the continuous flow of the mobile phase. It excludes bigger molecules as compared to the smaller pore size. They therefore pass through the column with the speed of the mobile phase and no retentions occur. The smaller molecules penetrate the pores where they are slowed down and taking longer inside the column. Consequently, some time will be made available to the molecules that could penetrate inside the gel, yielding them protection against the moving phase. Medium-sized molecules enter the pores in different capacity based on their size and shape. This causes the separation in the order of decreasing size. SEC makes no chemical interaction but is based solely on the physical size. Hence, it is ideal with proteins, polymers, and other biomolecules.
First, we will look at the clinical application of site exclusion chromatography, or SEC. SEC is used for the detection of protein aggregate in biopharmaceutical drug. It also helps in the removal of contaminant like SAR or small molecule from therapeutic protein preparation. An example of this is ensuring that monoclonal antibodies are pure and stable before injection. In addition to this, SEC is also used to purify important protein-like antibodies and enzymes in hospital and clinical laboratories. It can remove inorganic salts such as sodium chloride and small ions from biological samples, a process called deselecting. This makes SEC a very important tool in producing safe and effective biopharmaceuticals, especially those made from living cells. Next, in the pharmaceutical field, SEC is used to analyze the molecular size distribution of synthetic drug products. It also helps to monitor drug stability by detection, degradation, or aggregation over time. For example, it is used to confirm if a vaccine protein remains intact during storage. SEC can separate hormone, peptide, and vaccine protein based on their size to ensure the quality of the drug. It is also a reliable method to determine the molecular weight and stability of pharmaceutical products using calibrated check column. Lastly, SEC also has valuable use in environmental and food science. It is used to analyze macromolecular content in food additives and to study polysaccharide site in food thickeners and chilling agents. An example is measuring consistency in plant-based food sterilizer. Beyond that, SEC is used to study natural organic matter, or NON, found in water sample, which is important in environmental research. In the food industry, SEC helps to separate and study plant-based gum, polysaccharide, and sugar, which are essential for controlling food texture and quality.